Hello friends, myself Dr. Akhilesh, your physiology faculty. In this video, I will discuss the important points on nerve physiology. And the first topic which I will cover will be the types of neurons. What are the various types of the neurons? Neurons are of 4 types based on the number of processes coming out from cell body. So, based on the number of processes coming out from the cell body, so number of processes coming out of cell body, the neurons, they can be, one is unipolar neurons, in which one process will come out from the cell body. So this is the diagram of the cell body. The cell body is also called as soma. So it is also called as soma. So if, and this is the nucleus of the cell body, and if one process will come out from the cell body, so such type of neurons, they are called as unipolar neurons. And what are the example of unipolar neurons? So example is the neurons present in invertebrates, they are example of unipolar neurons. So the neurons present in invertebrates, they are example of unipolar neurons. The second type of neurons, they are bipolar neurons. So bipolar neurons means the two processes will come out from the cell body. So if this is the cell body, also called as soma, so if the two processes they will come out from the cell body, so this is called as bipolar neuron. Then the example of the bipolar neurons are the retina. The retina has the bipolar neurons. Then the third type of neurons are, they are the multipolar neurons. Multipolar neurons means many processes will come out from the cell body. So suppose this is the cell body or the soma. From the soma, the many processes are coming out. So this is an example of the multipolar neurons. So these multipolar neurons, they are present in our body, and the most of the neurons in our body, they are multipolar neurons. So most of the neurons in our body, they are multipolar neurons. For example, the motor neurons in the body, they are multipolar. The sensory neurons in the body, they are also multipolar. Then the Purkinje cells in the cerebellum, they are also example of the multipolar neurons. Then the fourth type of neuron is pseudo-unipolar neurons. In this neuron, one process will come out from the cell body, and it will give appearance that the two processes are coming out from the cell body. So this is called as pseudo-unipolar neurons. And the pseudo-unipolar neurons, they are present in dorsal root ganglion. So the dorsal root ganglion, they have the pseudo-unipolar neurons. So out of these 4 types of neurons, the very frequently asked is the bipolar neurons. So remember, the bipolar neurons, they are present in the retina. So they are the very frequently asked in the exam. Then after this, come to the another topic of the nerve physiology, that is the parts of the typical spinal motor neuron. So typical spinal motor neurons, they have number one, the cell body. The cell body is also called as the soma. The second part is the dendrites. The dendrites, they are the receptive process of the neuron. And the third part is the axon. Right. This axon, the, the first few micrometer of the axon do not have the myelin sheath, that is called as the initial segment. And it has the ending, which is also called as the nerve ending or the terminal button. So coming to the first part here, the first part is the cell body, also called as soma. So in the cell body, if I draw a diagram, so this is, look at the diagram here. So this I am drawing the diagram showing the dendrites and the structure of the cell body also. So this is the structure of the cell body or the soma, and here N stands for nucleus. So this one is the nucleus of the cell body, and this structure is the cell body or the soma. Right. Then the thickened part of the cell body. This is the thickened part of the cell body. This thickened part of cell body is called as axon hillock. So axon hillock is the thickened part of the cell body. Then these processes which are coming out from the cell body, these are called as dendrites. So these are called as dendrites. So this, the typical spinal motor neuron, is an example of multipolar neuron. So these are dendrites. Right. In the cell body, there are fine dust, there are the structures which are called as nisal granules. These nisal granules, they are also present in the dendrites. But these nisal granules, which are also called the tigroid bodies, they are not present in the axon hillock. So nisal granules, they are also called as tigroid bodies. So they are what actually? So they are the rough endoplasmic reticulum of the neuron. And what is the function of the rough endoplasmic reticulum? The rough endoplasmic reticulum, they synthesize protein, and here these rough endoplasmic reticulum, that is the nisal granules, they synthesize the neurotransmitter of the neuron. So they will synthesize neurotransmitter of the neuron. They have one more importance also. Whenever the neuron or nerve is in stress or there is nerve injury, these nisal granules, they will break down into fine sand-like structure. So this breakdown of the nisal granules into fine dust-like structure is called as chromatolysis. And chromatolysis occurs when the neuron is in stress or there is neuron or nerve injury. So let us write this. What is meant by the chromatolysis? So chromatolysis means it is the breakdown of nisal granules into fine dust. So breakdown of nisal granules into fine dust. And when this occurs when the neuron is in stress or there is nerve or neuron injury. So this chromatolysis occurs when the neuron is in stress, it is in stress, or there is injury to this neuron. Right. So this is the importance of the missile granules or hygroid bodies. Then the cell body or the soma, it will continue into the axon. So suppose if I draw the diagram of the axon, so you can see here. So now this is the structure showing the axon. The first few micrometer of the axon do not have the myelin sheath, and that is called as what? So that is called as the initial segment of the axon. So this one is the initial segment of the axon. Right. And on the axon, there are myelin sheaths. So these myelin sheaths are formed by the Schwann cells in case of the spinal motor neuron. So this cell forming the myelin sheath is the Schwann cells. So these Schwann cells, they are forming the myelin sheath over this neuron. Right. Then this axon will end into the terminal button. So this is the structure of terminal buttons where the vesicles containing neurotransmitters are stored. So neurotransmitters are stored where? They are stored in the nerve terminal, in the terminal buttons. So these are the terminal buttons containing neurotransmitters. So neurotransmitters are stored where? They are stored in the nerve terminal, right? So coming to these parts of this spinal motor neuron, so one part I told you it is the cell body or the soma. The second part is the dendrites. The third part is the axon. So this is the structure of the axon. So this is the third part of the spinal motor neuron. So I have told you about the nuclear granules which are present in the cell body. These nuclear granules, they are present surrounding the nucleus, right? So they are also called as the structures present surrounding the nucleus. Surrounding the nucleus is called as perikaryon. So they are present surrounding the nucleus, but they are absent in the axon hillock. So present surrounding nucleus, this is also called as perikaryon. Peri means surrounding, karyon means nucleus. So it is also called as perikaryon. Perikaryon means they are present surrounding the nucleus, but they are absent in where? They are absent in the axon hillock. Right. So they are absent in the axon hillock. Now come to the second part of the spinal motor neuron, that is the dendrite. So these dendrites, which I have shown in the diagram, they are the receptive process of the neuron. What is meant by that? So receptive process of neurons means they will receive the information from other neurons, because these dendrites, they are the part of the synapse. So the most common synapse is the dendro plus dendrite plus axon. This is also called as exodendritic synapse. So most common synapse is exodendritic synapse. So the dendrite of one neuron, it will receive the information from the other neuron, and it will send this impulse towards the cell body. So this is the one importance of the dendrite, right? The second importance of dendrite is that these dendrites, They are the site of generation of electrical activity in the neuron. And the third one is that they have the missile granules in them, so they can synthesize proteins, and these proteins are these proteins play a role in learning and memory. So coming to this, the dendrite. If I draw the diagram again of the dendrite, so these are the dendrites. So just now I told you that these dendrites, they are what? They are the receptive process of the neuron. So they will receive the information from the other neuron and transmit it towards the cell body. So I will write here, so number one is they are the receptive process of neuron. Means they will receive the information from other neuron and it will transmit this information towards the cell body. Second thing is that they are the site of generation of electrical activity of the neuron. This electrical activity is called as electroencephalogram. So they are site of generation of electroencephalogram. So electroencephalogram is the electrical activity produced in the neuron, right? Then the third one is that these dendrites, they have missile granules in them, and these missile granules, they will synthesize the proteins, right? And these proteins, they play a role in the learning and memory. What happens is, whenever the neuron is stimulated, it will generate the electrical activity and it will excite the dendrite. When the dendrite is excited, the missile granules, they will start synthesizing the protein, and this protein will be uh, play a role in learning and memory. So dendrites, they play a role in learning and memory. So they will play a role in learning and memory. So remember these 3 points on the dendrite. Number one is receptive process of neuron. Number two is their site of generation of electrical activity of the neurons, that is the electroencephalogram, and they play a role in learning and memory because they have missile granules, and these missile granules, they can synthesize proteins, and these proteins, they play a role in learning and memory. The third part of the neuron is axon. So this axon, the first few micrometer of the axon is called as initial segment. So it is the first few micrometer of the neuron, and this part is what? It is unmyelinated part of the neuron. And what is the importance of this initial segment? That this initial segment is the site of generation of action potential, the site of generation of the action potential in the spinal motor neuron, the site of generation of action potential in spinal motor neuron. Right. Now the question is, why it is a site of generation of action potential? The answer is because it has the lowest threshold of excitation. So it has the lowest threshold of excitation. Now the another question is again here, that why it has the lowest threshold of excitation. So again answer is because it has the large amount of the voltage gated sodium channels. So because it has large number of these sodium channels. The sodium channels, they are present in very large number in the initial segment, right? Then if I draw the diagram, then this is the right, then this is the part of the cell body, the thickened part of the cell body. If this I level it, it is the thickened part of the cell body called a what? Axon hillock, right? Then after the axon hillock, what will start here? So after this, the axon will start, and the first few micrometer of the axon is called as the initial segment. So this is called as the initial segment, and this axon, it has the myelin sheath over it. So this myelin sheath is formed by the Schwann cells in case of these spinal motor neurons, right? So these are the Schwann cells forming the myelin sheath over this neuron, right? So in this myelin sheath, you can see that there are periodic constrictions in the myelin sheath. So these periodic constrictions, they are called as the nodes of Ranvier, right? So what are the nodes of Ranvier? The nodes of Ranvier, they are the periodic constrictions in the myelin sheath. So they are the periodic constrictions in the myelin sheath, and this myelin sheath is present where? This myelin sheath is present over the axon. So remember, in the exam it is asked that where are the nodes of Ranvier present? So answer is on the axon, right? If I draw the diagram again, so suppose this is the diagram showing the axon, and this is the myelin sheath formed by the Schwann cell. The Schwann cell is forming the myelin sheath here, and these periodic constrictions, they are called as the node of Ranvier. So here I have shown, so these are the nodes of Ranvier, right? So these nodes of Ranvier, their width is 1 micrometer. So you have to remember the 2 points over this. What is the width of this node of Ranvier? The width is 1 micrometer, and the distance between the two nodes of Ranvier, which is also called as internodal distance. This internodal distance or distance between two nodes of Ranvier is 1 millimeter, right? What is the width of the nodes of Ranvier? The width is 1 micrometer, and what is the distance between two nodes of Ranvier? This is also called as the internodal distance. This is equals to 1 millimeter. And the nodes of Ranvier, they have the one important property also, that they have the highest density of voltage gated sodium channels. The nodes of Ranvier, they have the highest density of these sodium channels, right? The sodium channels, their highest density on the nodes of Ranvier, right? And also know one more thing about the node of Ranvier, that the first node of Ranvier is the site of generation of action potential in the sensory neuron. So in the sensory neuron, the site of generation of action potential is first node of Ranvier. But in the motor neuron, it is the initial segment of the axon, which is the site of generation of action potential. Remember here that the node of Ranvier, so in the sensory neurons, the site of generation of action potential is, so action potential is generated on the first node of Ranvier, right? And just in previous section, I told you about in the motor neuron, the action potential generated on initial segment. So remember the following points on the node of Ranvier. Then where are the neurotransmitters stored? The neurotransmitters are stored in the terminal buttons of the end of the axon, right? So these are the parts of the typical spinal motor neuron. Now coming to the another topic of the nerve physiology, that is the glial cells. The glial cells are also called as neuroglia. These cells, they will support the neurons in the central nervous system, and these glial cells, they can divide. So they can divide. So neuroglial cells or the glial cells, they can divide, but the neurons, they do not divide, except uh, um, exceptions are there, except the few neurons of hippocampus and neurons of the olfactory epithelium, which can divide. So other neurons, they do not divide. So remember, the other neurons, they do not divide, but some neurons of the hippocampus and neurons of the olfactory epithelium, they can divide. But the glial cells or the neuroglial cells, they can divide. So if you read the tumors of the central nervous system, so most of the tumors you will read that they will originate from the glial cells or the neuroglia. Then what are the types of the glial cells? So these glial cells, they are of 5 types. What are their names? One is called as microglia. Second one is called as astrocytes. Third one is called as oligodendrocytes. The fourth one is called as the Schwann cells. And the fifth one is called as ependymal cells. Right. So these are the 5 types of the glial cells. One is microglia, another one is astrocyte, third one is oligodendrocyte, fourth one is Schwann cell, and the fifth one is the ependymal cells. Right. So coming to the first one, that is the microglia. The microglia word begins with the alphabet M. Remember the 4M about the microglia. The first M is what? M stands for macrophages. So microglia, they are, so the microglia, they are the macrophages of the central nervous system. So first M stands for macrophages. The second M stands for monocytes. Monocytes means the macrophages, they are derived from which WBC? The macrophages are derived from monocytes. So therefore, the microglia, they are also derived from monocytes. So the microglia, they are derived from the monocytes. The third M is for the marrow. Marrow means bone marrow. The monocytes, they are produced in the bone marrow. So therefore, these microglia, they are produced in the bone marrow. So they are produced in bone marrow. And the fourth M is mesoderm or mesodermal in origin. So during the embryonic development, the microglia, they originate from the mesoderm or they develop from the mesoderm. So these microglia, they are mesodermal in origin, right? So remember the 4M about the microglia. One is M stands for macrophages of central nervous system. Another M stands for what? Another M stands for the monocytes, right? The third M stands for the marrow, and the fourth M stands for mesodermal in origin. So now it is also very frequently asked in the exam that what are the macrophages of liver. So you know very well the macrophages of the liver, they are the Kupffer cells. So Kupffer cells, they are the macrophages of the liver. Then after the microglia, the second neuroglial cells are the astrocytes. These astrocytes, they help in formation of blood-brain barrier. So they will help in the formation of blood-brain barrier. So this is their one function. They will help in the formation of blood-brain barrier. Their second function is they will maintain the brain ECF. So they will help to maintain the composition of brain ECF. To maintain the composition of brain ECF, and what they do, that they will uptake the potassium and the glutamate or glutamic acid from the brain ECF, and what they will do, that they will convert the glutamate into glutamic acid. So remember this fourth one, that they will convert the glutamic acid into, or the glutamate into glutamine. The glutamic acid or glutamate is the main excitatory neurotransmitter in the central nervous system. So they will convert this excitatory neurotransmitter glutamate into glutamine. Then the third glial cells is oligodendrocytes. So these oligodendrocytes, they help in formation of the myelin sheath in the central nervous system. So they will form the myelin sheath in the central nervous system, and one oligodendrocyte, it will form the myelin sheath around 15 to 20 neighboring axons in the central nervous system. And the Schwann cells, they will form myelin sheath in the peripheral nervous system. Right. So how we will remember that? The oligodendrocyte, they will form myelin sheath in central nervous system. So C for central nervous system, then you can make this C as O. O stands for what? O stands for oligodendrocyte. So oligodendrocyte, they will form myelin sheath in the central nervous system, and Schwann cell in the peripheral nervous system. Then ependymal cell, remember ependymal cell begins with what? EP. E, right? So this ependymal cells. So these ependymal cells, they will form the epithelial lining around the ventricles in the central nervous system. So they will form the epithelial lining surrounding the ventricles in the central nervous system. Right. So epithelial lining. So epithelial lining means what? EP. EP for epithelial lining and EP for ependymal cells. So these ependymal cells, they will form the epithelial lining surrounding the ventricles in the central nervous system. Right. So these are the important points on the neuroglial cells, or which are also called either. So these neuroglial cells, they are also called as the glial cells, which are of 5 types. Right. Microglia, they are macrophages of central nervous system. Astrocytes help in formation of blood-brain barrier. Oligodendrocytes, they will form the myelin sheath in central nervous system. And the Schwann cells, they will form the myelin sheath in peripheral nervous system. And the ependymal cells, they will form the epithelial lining surrounding the ventricles in the central nervous system. So in the next part of this, the glial cells, I will discuss about the two applied aspects of these glial cells. Number one applied aspect is the multiple sclerosis, and the second one is the Guillain-Barré syndrome. So multiple sclerosis, this is an autoimmune disease. In this autoimmune disease, what happens is that there is patchy loss of the myelin sheath in central nervous system. There is patchy loss of myelin sheath in central nervous system, and this patchy loss of myelin sheath is due to the lesion or damage to the oligodendrocyte. So you have to remember the cell name involved in the multiple sclerosis. The cell name is what? Oligodendrocyte, because the oligodendrocytes, they form the myelin sheath in the central nervous system. Right. Then the second applied aspect is the Guillain-Barré syndrome. This is the corresponding part of the multiple sclerosis in the peripheral nervous system. So here what happens is there is the patchy loss of myelin sheath in the peripheral nervous system. So there is patchy loss of myelin sheath in the peripheral nervous system, and this patchy loss in the peripheral nervous system is due to lesion or damage of which glial cell? That is the Schwann cells, because the Schwann cells, they will form the myelin sheath in the peripheral nervous system. So this is due to lesion or damage of the Schwann cells, because the Schwann cells, they will form the myelin sheath in the peripheral nervous system. So remember these two applied aspects on this neuroglial cells, right? Then in the next video, I will discuss about another important topic from the nerve physiology, that is the equilibrium potential, resting membrane potential, and the action potential, and the other remaining parts of the nerve physiology. So this much in this video. Thank you very much for watching this video.